these are the various ion channels, the sodium channels, the potassium channels, and the sodium-potassium pump, along with various other channels which are present on the membrane. So the resting membrane potential at the resting phase arises as a combination of the various types of protein channels or ion channels. So what is this exact combination? So this is the sodium channel. The sodium channel actually is structurally quite complex. It has various types of subunits, and subunits have their own individual segments and so on. But if you look at it through a very superficial aspect, we can see that it has an activation gate, an inactivation gate, and these control the influx or efflux of sodium ions. And then we come to the potassium channel. So the potassium ion channel consists of a particular selectivity filter. This is a selectivity filter, and this selectivity filter is lined by certain carbonyl oxygens. And these carbonyl oxygens are arranged in such a way that these interact with only specific hydrated potassium ions, such that when these enter the selectivity filter, it leaves the water molecule and the dehydrated potassium molecule enters the cell. However, sodium molecules cannot pass through this because the carbonyl oxygens are too far apart for the sodium molecules to actually interact with them. So then we have the sodium potassium pump. A pump uses energy to drive ions against their concentration gradient in order to maintain a certain equilibrium. So the movement of ions will be something like 3 sodium molecules exit the cell and 2 potassium molecules enter the cell, and 1 ATP molecule is broken down in order to make this happen. So outside this is inside. So we have talked about the contributing factors for the resting membrane potential. That a combination of these ion channels help in actually contributing to the minus 70 or minus 90 millivolt, that is the resting membrane potential of a particular cell. For skeletal muscle, it's around minus 90 millivolt, and for neurons or nerve muscles, it is around minus 70 millivolt. So we'll use a trial combination method for understanding or determining the factors that actually influence or contribute to the resting membrane potential value. First, we need to know what do we actually use to derive the membrane potential. So we've talked about the Nernst potential for deriving the equilibrium potential of the membrane, which is minus 61 by the charge log of concentration inside by concentration outside, or outside inside you can omit the negative sign. But this actually, since the concentration gradient is not the sole factor for contributing to the resting membrane potential, because our physiological system is arranged in such a way that the membrane is 100 times more permeable to potassium ions than sodium ions. Okay, so along with other specific ion channels and pumps, so the membrane is selectively permeable for some specific ions. So we need to consider the concentration gradient as well as the permeability. So while calculating the membrane potential, we'll use another variable, which is permeability. Equation. This is the Goldman equation. Now, what is this combination that actually affects the resting membrane potential? So say suppose the only factor contributing to the membrane potential was diffusion. So according to the gradients, say suppose this is a rough diagram of a potassium and sodium channel. So say suppose this is a potassium channel, say suppose this is a sodium channel. According to the gradients, potassium would move outside, sodium is almost the reverse, so move in. We know the equilibrium potentials of both these two ions, and we can calculate an overall potential. Like, since this is actually a summation, use both the values in this equation itself, so it comes around minus 86 millivolt. the membrane potential. It's very different from what we know to be the resting membrane potential. So, see, suppose we have another factor which is a sodium potassium pump. So, in this case, these are the same channels. And the pump uses movement in the opposite direction. So there is actually more efflux of positive ions compared to the influx of positive ions. So there is approximate negativity which happens to apparently develop solely because of this. So if you take a combination of these ion channels, the potential comes to almost around minus 90 millivolt skeletal muscle and minus 70 millivolt for neuron. Now the membrane is mostly impermeable to the large negative ions. So there is an unequal distribution which happens on both sides of the membrane that also contributes to the negativity inside. So this unequal distribution is called the Gibbs-Dunan equilibrium. So all these factors combined contribute to the minus 90 millivolt or minus 70 millivolt, which is the resting membrane potential of the cell. We can approximate that the sodium potassium pump contributes almost minus 4 millivolt to the resting membrane potential. Thank you.